Thank you so much for having me. It's really a pleasure to be here. So this is going to be a little different than some of the other talks because it's not about social science. It's about actually biological science, but I would say with a very social science lens. Okay, and this is joint work with my fantastic collaborator, Ryan Hill at Northwestern. Okay, so basic science, when I say the word basic science, I'm talking about the kinds of science that might advance our fundamental understanding of how the world works, but that in and of itself can't necessarily be commercialized or be profitable. However, economists think about advances in basic science as serving as key inputs into more applied science and economic growth. So it's an interesting, important area. And because you're not compensated directly with profits when you make a discovery in basic science, we think, people are compensated in some other way. How do we think about how they're compensated? We think that a really important piece of it is credit. This idea that people recognize that you are instrumental in bringing this thing into being is something that's really motivating for scientists. But how do you get credit? At least part of it seems to be about getting your findings out there first. So if it's really important to be first, it's not surprising that we've seen sort of across history, really sort of famous examples of people fighting with each other about who was the first person and who ought to get the credit. So, Newton and Leibniz are a famous example. They historically really didn't like each other. They both wanted to be thought of as the inventor of calculus. Charles Darwin and sort of the lesser known Alfred Wallace were actually both working on a theory of evolution at the same time. It was only when Darwin learned about Wallace that he really hurried up and published on the origin of species. So we think that this is something that really permeates science. And maybe, so then the sort of question we're interested in is how should we think about this incentive where it's really important to be first? You might think it's good for science. It might get people to work really hard. And it seems like it also encourages people to disclose their findings and put them out into the open because it's only by doing that that people can know you were first. So those are all good things. However, we were also interested in this idea that there may be a dark side or potentially multiple dark sides. But what we really want to focus on in this talk is actually something that Skip, I think, alluded to in the opening remarks. People might, in their pursuit to publish first, cut corners on quality. So that's really what this paper in this project is about. Do we think that this is a problem? And here's actually a quote I just really like because it's a scientist in his own words sort of saying this better than I could. So Svante Pääbo is an evolutionary geneticist. He actually won the Nobel Prize a year or two ago. And part of his work was trying to sequence the Neanderthal genome. And he has this quote from his book where he talks about a competitor of his, somebody named Hendrik. And he said, Hendrik's paper illustrated a dilemma in science. Doing all the analyses and experiments necessary to tell the complete story leaves you vulnerable to being beaten to the press. Even when you published the better paper, you were seen as mopping up the details after someone else who made the real breakthrough. And this quote highlights the tension that we want to think about in this paper, which is that on the one hand, when you spend longer, you can do a more careful job. And most scientists, they want to do that. But they realize that in spending that additional time to do the really careful work, they also increase the risk that somebody else sweeps in and preempts them. And that's going to be a force pushing them perhaps in the opposite direction. Okay. So in this project, we're going to focus actually on a very particular field of science called structural biology. So I want to talk a little bit about that and why we made that decision. So structural biology, for those of you who are not familiar, we were not familiar when we started this project, is the study of the 3D structure of macromolecules. For the most part, these macromolecules are proteins. So scientists in this field are trying to figure out the 3D shape of proteins. One reason to study this, it sounds sort of niche, but it's actually a really important field of science. So what you see on the left here is actually an image from the journal Science. And this image you see here is a 3D model of the COVID-19 spike protein. So at this point, probably we've all seen the picture of the COVID-19 virus, this sphere covered in little spikes. Those little spikes is that protein. And your body needs to learn how to recognize that protein in order to mount its immune response. So actually, this structure was really critical in the vaccine development efforts. We needed to teach the immune system to recognize this. So that's one reason to focus on it. I think over a dozen Nobel Prizes have been awarded in this field. But for our purposes, the data in this field, just sort of talking to a room of people who care about open science, and happy in the Q&A to talk a little bit more about the infrastructure this field is set up. But the data in this field is just very amazing. So there is this resource called the Protein Data Bank that is a very complete record of all of these 3D structures. And in the Protein Data Bank, we can learn about things like objective measures of the project's quality. So how well done is this protein structure? That's like a pretty unique thing. You could imagine in, let's say, the field of economics, how would you measure how well done a paper is? You could count its citations, but that's going to mix up how well done the project is with just maybe how interesting the question is. We want to separate the two. You could imagine looking at sample sizes, perhaps, in experimental fields, but this is just I'm going to talk more about this. This was very unique about this field. There's also information on the project timelines, which lets us know who was working on which projects and when. There's links to the publications that debut the protein structure and other just incredible details about these projects. Okay, so now let me just briefly talk about a sort of theoretical model of how competition and quality might interact in science. And then what I'll do from there is I'll talk about the data that we have and how we can link sort of the predictions that come from this model to actual things we can really measure in the data. And then with both of those pieces in hand, I'll show you some results. Okay, so the model we have in mind is that projects, before anyone started them, vary in what we call their ex-ante potential. So in this setting, that would mean sort of there's a bunch of proteins as a scientist you could try to work on. Some of them, like the COVID-19 spike protein, this is an incredibly important protein. If you solve this, there's going to be a lot of interest. It's going to publish in a top journal. Other proteins, some protein that is in a zebrafish and we don't even know its function, you might know, yeah, I could do this project, but it's not going to really publish super well. So we would call those two proteins, the first one would be a high potential protein, and the second would be call it a low potential protein. Okay, so scientists, when they're working on a problem, they decide how long they want to spend working on it. We call this the time they let the project mature for. And when they're making this decision, it's exactly that Svante Pabo trade-off he was talking about. You can spend longer and you can do a better, more careful job. But the longer you spend, the greater is the probability that somebody else, if they're working on it, will beat you to publish. So you're making that trade-off. The key ingredient, though, is that it's not random which projects people work on. And actually, right, if we're all of us deciding which proteins we want to work on, the majority of us would want to work on the COVID-19 spike protein, and only a small minority would want to work on the random zebrafish protein. So that's what's actually going to sort of make this whole model turn here. We're all going to collectively, even if we don't talk to each other, we're all going to understand that if we chose the COVID-19 spike protein, lots of other people are working on that. So the people who choose that protein are going to also realize it's very competitive. If I want to be first, I need to really rush. And so actually the key prediction that comes out of this is that sort of perversely, it's the most important projects that we think are going to be done the most rushed with the most corners cut. That's the prediction that we're going to try to take to the data in this field of structural biology and see if it looks like it's the case. Okay, so this is just outlining the three things we're going to want to try to test. First, high potential projects should be more attractive to enter. So it should be those projects where we see a lot of people working at the same time. Our model clarifies that when you perceive lots of other people working on it, the scoop cost more quickly outweighs the improved quality benefit. And so it's those projects where people cut themselves short the most. And if you put those together, you get this key prediction, which is actually, it's the most important projects that should be done the most badly. Okay, so you can I think this is somewhat straightforward to think about conceptually. Thinking about testing this is harder. And so this is where the field of structural biology for our purposes really shines. And I think was the really fun part about this project. And it just kind of shows you how good data collection can just be so useful, maybe in ways that the data collectors themselves did not anticipate. Okay, so let me just give you a little bit more background on structural biology and how this process works. So 90% of the protein structures, so think like that model of the spike protein I showed you, 90% of those are solved using a method called X-ray crystallography, which broadly involves 3 steps. The first thing you need to do is get a super saturated solution of your protein and slowly evaporate the water off and try to grow a protein crystal. So if you've ever grown like a salt or a sugar crystal in your kitchen, it's not conceptually that different, but it's actually just very tedious. So in this, it can be very hard to get these proteins to crystallize. Scientists are sometimes, trying hundreds of different combinations of experimental conditions. So they'll vary things like the temperature, the pH, or other things they add. So they spend a lot of time working on this phase. But once they have a protein crystal in hand, they can take it to something called the synchrotron facility, and they will beam this protein with X-rays, and that's going to produce something called the diffraction pattern, which you can maybe see on the right side of the screen, that little white square with a bunch of black speckles. So that's like the data. And then what they're trying to do is they're trying to reverse engineer what must the 3D structure of that protein have been that would have generated a diffraction pattern that looks like that. So they'll start with some crude model, or maybe they'll take a model of a similar protein off the shelf, and they'll say, okay, well, if that was really the true model, what would the diffraction pattern look like? And then they'll compare that to their experimental data, and the two won't match. So then they'll go back to their model, they'll tweak, and they'll repeat. And this repeated process is called refinement. And in this stage, they can really do more rounds of refinement, trying to improve their model and make it better and better. And we think of that as being pretty akin to the maturation phase in this model. The more time they spend tinkering with their model, the better the model will be, but they may feel the pressure to hurry up. Okay. So then what is the Protein Data Bank? So the Protein Data Bank was established in 1971, and it's a database for the 3D structures of these large biological molecules. The story of sort of how it was started is pretty amazing. I won't get into the details, but basically there was a PhD student in this field in the 70s, just as sort of the number of structures was starting to take off, who just felt that it was incredibly important to be getting these into a centralized place. And she basically, I think to a large extent, left her work at the lab bench in order to start this, and really was faced with a lot of headwinds in doing so. Individual scientists were not chomping at the bit to sort of be putting their work into this repository. But she, over the years, gained traction, and then a big change happened in the 1990s when basically all scientific journals agreed that they would not publish a structural biology paper unless that structure was uploaded to the Protein Data Bank. And it really took that kind of a coordinated effort across the field for this data bank to take off. Today, the Protein Data Bank contains, actually, this slide is outdated. It contains, I think at this point now, nearly 200,000 protein structures. And so it's just been an incredible resource for researchers in this field. And if any of you are following the interesting AI developments that are happening in this field with AlphaFold and DeepMind, I mean, this was an incredible resource for those AI labs and firms to build their predictive models. So just an unbelievable resource. Pharmaceutical firms are heavy users of these protein structures. It's very valuable. Okay, so if anyone's interested and has their laptop with them, I mean, you can just log on to the Protein Data Bank's website. It's free, it's open access. They have programmatic APIs to let people download the data. If you look for PDB ID 4CMP, you will stumble across this structure, which is, a particularly interesting structure. It's the CRISPR-Cas9 protein. So it's a really critical protein in this CRISPR-Cas9 gene editing system. And you get to kind of get a sense of some of the things that are contained in this data. So there's the 3D structure of the protein. You can see in small print up there, the deposit date and the release date. So this is the date it was first placed into the Protein Data Bank. When you first place it in there, it's actually kept hidden from the public. Then there's the release date. That corresponds to when the paper was published, and all of a sudden your deposit is available to see by the public. So those dates are going to be useful for us. You can see the authors, including the last author here is Jennifer Doudna. You can see these measures below in this graph of the structure's quality. I'll talk more about that in a second. And then it links via a PubMed ID to the paper that sort of is debuting this protein structure. Okay. So now let me talk about how we're able to measure quality in this setting. So there's actually several different measures. The Protein Data Bank has been very proactive. They want these quality measures because what they want is people who are not experts in the field of structural biology to be able to look at these structures and say, I'm not an expert, but is this reliable and can I use it as an input into my own work? So they, in some cases, have developed these measures, or at least they independently validate them. So the first measure is something called refinement resolution, which you can really think of as being very similar to the resolution of a photograph. So this picture is just showing you actually the same protein structure, but as you move to the right, it's been solved at increasingly better levels of refinement resolution, and just much more detail emerges at better refinement levels. The next measure is something called R-free, which is meant as a measure of model fit. It's how well does the model you generated match up with the experimental data. And then finally, there's something called the Ramachandran outliers. So we sort of understand from chemistry and physics that certain atoms can only bind at certain angles. And the protein data bank can actually go through your proposed structure bond by bond and just flag instances where things are bound at sort of chemically infeasible angles and then report how frequent of an occurrence that is. So what I'll just do in the rest of the talk to sort of make things simple is just combine these three measures into a single index where higher scores correspond to better quality and the units will be in standard deviations. Okay, the next thing we'd like to be able to see is how long the scientists actually spend working on the project. And because we have these different dates associated with the projects, we can do that to some extent. It's not perfect. We don't get to see the date when they first start growing the protein crystals. But once they have the protein crystal and they take it to a synchrotron, that date is collected for the most part. It's called the collection date. So we can see that date, and then we can see the date where they deposit the structure in the protein data bank. So we think what's going on during this time period is the scientist is solving the protein structure, potentially writing the draft of their paper, and then uploading. So that's like a sort of, we think, a reasonable proxy for the maturation period. It takes about 1.5 years on average to do all of those steps. Okay. The next thing we would like to be able to measure is competition. Were there many people working on the same protein at the same time? And we can do that actually pretty well. So we can use, basically the protein data bank can help you find they can tell you how close in protein space two proteins are to each other. So we can find proteins that are identical or nearly identical. And because we have the dates, we can figure out if they were being worked on concurrently. And that lets us construct basically an indicator variable for, does it look like there was a race going on for this protein or was really only one person working on it? It's not perfect. Some people may abandon their projects and we don't know, but it's we think a pretty good proxy. And then the last thing that we really need to be able to measure is this idea of project potential. How important did scientists perceive this project to be? Now, we have to construct this measurement ourselves, but what we do is we use really detailed characteristics of the proteins. So things like, what is the protein's function? The PDB classifies them into things like oxygen transport proteins, membrane proteins. We can use the organism they come from, whether it's a human or a monkey or a mouse. And actually, very interestingly, we can even count the number of papers that were written about this protein prior to anyone solving the structure as sort of yet another measure of maybe sort of pent-up demand for this protein structure. So we can use these characteristics to try to predict the number of citations we think it's going to get, and we use that predicted value as our measure of potential. So that actually gives us all of the pieces we need now to try to start testing the predictions of this model. So the first prediction is that these projects we code as being high potential ought to be more competitive. And that's what this figure is showing you. There's a positive association between potential and the probability that we observe some sort of priority race, multiple people working on this protein at the same time. So I think the way that I found it helpful to think about effect sizes here is to think about, imagine a protein that is in the 90th percentile of this potential distribution we've constructed to one that's in the 10th percentile. So you can just sort of imagine we're comparing a high potential protein to a low potential protein. And if you do that 90-10 comparison, you can see that these high potential proteins are 4 percentage points more likely to be in a priority race, which maybe doesn't sound that big, but sort of the baseline probability is about 8%. So it's a 50% increase in the likelihood that you face a competitor. Okay. And then the next thing we want to test is, okay, so these high potential proteins are more competitive. Are they completed faster? And this graph is showing you yes. Using that same 90-10 comparison, high potential proteins are finished 2 to 3 months faster, again, off of a mean of about 1.5 years. And this in turn seems to translate to lower quality. These high potential structures are lower quality. About using this 90-10 comparison again, 0.6 to 0.7 standard deviations lower in quality. So it seems like, this is consistent with a story where people are rushing because they don't want to be scooped, and that's harming the quality of the work that they're doing. But one thing that when we look at this graph, we want to be very careful about in the interpretation is that, is this really all being driven by rushing and the fear of being scooped? Or is there something else that's just correlated with potential that could be driving these results? And in particular, a very reasonable concern could be that high potential proteins are just harder to solve. And so nobody's rushing. They're just going to be lower quality because it's a harder problem. So that's something that we really wanted to take seriously because that's a very different story with very different sort of policy implications. So we did a couple of things. I want to tell you about one of the things that we tried. So, okay. So actually, sorry, I'll tell you about, I take that back. I'll tell you about two of the things we tried. So the first thing that we tried to do is just control directly for how complicated these proteins are. We talked to a lot of structural biologists, and one thing that they told us that was sort of reassuring is that it's actually, from their perspective, very hard before they start to know which projects are going to be easy and which projects are going to be hard. It comes down to crystallizing the protein, which just seems to be very unpredictable. Some proteins crystallize beautifully, others are really hard, and they don't know before they start. So the fact that the scientists kind of can't tell gave us confidence that maybe it's not correlated with things that are easy to measure, which is what our potential measure is comprised of. So they sort of told us that, but they said big proteins are on average harder. They just tend to be harder to crystallize. And then even once you crystallize them, you're solving for a more complicated model. So the protein data bank has very nice different measures of a protein size. And so we can control for those. So what happens when we control for those? So in panel A, I'm showing you in this table, I'm actually showing you all three of the different quality measures plus the quality index on the far right. So we can just focus on the quality index for now. In panel A, I don't control for the project's complexity, and in panel B, I add those controls. And two things sort of jumped out to us. One is that the R squared goes up a lot when you control for this complexity. So that suggests to us that these controls that we're choosing, they are very predictive of the project's quality. But the coefficient on potential doesn't change very much, which suggests that yes, these complexity measures are very predictive of quality, but they're just not that correlated with project potential. So there can be complicated proteins that are high potential. There can be simple proteins that are high potential. And this sort of squares with our understanding of the field, where there's lots of proteins that we haven't solved. How do we decide what's exciting? It actually depends on what's going on in other fields. So a cellular biologist will say, oh, I've realized that this new protein is very related to overexpression of this protein seems like it has implications for cancer. All of a sudden now that protein becomes a very high potential protein. But those types of discoveries we think seem to be uncorrelated with how hard it's not the case that there's just only hard proteins left. So that, I think, conversations with structural biologists were very useful here. We still were not totally convinced, so we tried one other thing. We learned about something called the Protein Structure Initiative and the Structural Genomics Consortia. So there are different types of institutions that structural biologists belong to. A lot of them are university researchers. That's probably what we all have in mind. They're the ones who sort of feel this publisher perish pressure, and they're very competitive. But about 20% of the structures in our sample were deposited by researchers who were part of these structural genomics consortia. These were publicly funded efforts, very much in the spirit of the Human Genome Project, where they were just trying to solve as many proteins as they could and deposit them in the protein data bank to get good coverage of the protein folding space. At one point, they thought they might be able to solve all proteins. They kind of realized that was going to be too ambitious, but they face very different incentives. They, most of the time, don't even publish a paper when they put these proteins in the protein data bank. So they're much less concerned with being first. They only publish about 20% of their projects, whereas the university researchers publish nearly 90%. So we have this interesting sort of control group, you might think, of researchers for whom being first just seems like it matters a lot less. And so if we can contrast the behavior between these two groups, you can kind of think it's like we're holding everything else fixed, except we're sort of dialing competition down. How does behavior change? So that's what I'll show you in these last two figures. So this is showing you the results for maturation. So in blue, you have the university researchers. In red, you have the structural genomics researchers. So the first thing that jumps out at this graph is just there's a big level difference between the two. We think at least part of that is because structural genomics researchers are not trying to write and publish papers, they deposit the structures immediately as soon as they have them, whereas the university researchers are writing papers and just, it might be, that could explain the level shift. But what I do want to draw your attention to here is the difference in slopes. The university researchers, as we've already seen, move more quickly when the projects are high potential. The structural genomics researchers, if anything, actually take longer on the more important projects. And the differences in these slopes is statistically significant at the 1% level. We can then look at how this impacts quality. And so again, in blue, we have the university researchers, and in red, we have the structural genomics researchers. Both groups, we see that there is a negative relationship between potential and quality, but it is significantly steeper for the university researchers. How you want to interpret this red line, I think different people could have different reasonable views. One could be that the structural genomics researchers are competitive to some extent. Maybe for the 20% of structures they're trying to publish, they do race a bit. That's reasonable. The other you could say is maybe that this is reflecting some sort of omitted variables bias that we cannot fully account for. But then I would say the difference in these slopes should be how much of this effect you ought to attribute to competition. And the difference there is statistically significant. So that's the last thing I'll show you. I'll just wrap up here by saying a few things. So first, I would say that I hope these results have convinced you that competition in science leads researchers to work faster and produce lower quality work. In the paper, we try to think more carefully about, are these effects big enough that they should matter? And the answer, I think, is a bit, it depends. Happy to talk more about that. And then, we've also done some work around people doing, how much does it cost the field to sort of fix this low quality work? We see people redepositing low quality structures. We estimate that this has been quite expensive, that we've spent between $2 and $5 billion in an effort to clean up the low quality work that's been done in this field. And then just lastly, to sort of say more broadly, I don't think this paper can definitively say sort of anything about the optimal level of competition in science and do we have too much or too little, because competition just surely affects science in so many ways that we have not considered here. One thing we've heard a lot about is how it might reduce collaboration and the free sharing of ideas. It could impact who wants to enter certain fields of science and who's deterred by that culture. But I think, and I hope that this just brings up interesting questions of alternative models we could have of science. So things like the Protein Structure Initiative and the Human Genome Project are examples of when we have people sort of collaborating and sharing more information and sharing credit rather than individual labs competing against each other. Which one is better? I don't know the answer, but I think it's a really important thing. And it's hopefully people in this room, it's a question people want to engage with. So, okay, I'm out of time. Is there still a little time for questions? Okay.
Hi, thank you very much for that presentation. I had maybe a very naive question, but when you're thinking about competition, the examples you showed in the beginning were like one individual against another individual, let's say for priority for calculus. But in the context of these projects, is there a concept of controlling for the number of collaborators for a project? Like, does that play a role? If it's one person who's trying to race against another versus a team of people racing against others?
That's a great question. Most of these projects involve teams. And one thing that we see is that the teams tend to be larger for the more competitive projects, which I think it makes a lot of sense. One way to try to win the race is to move faster. And you can move faster by just doing less stuff, but you can also try to bring more resources to the project. And we do see people doing that to some extent. Yeah.
Thanks. This is super interesting. It seems like instead of changing the model of science, another mechanism for addressing it would just be to increase the disincentive to quality. And so I was just wondering, given that you can observe quality so cleanly through this database, like, why isn't that already changing people's incentives?
I completely agree with you. You're like, people have said to us, oh, you've like cherry picked this field. This is the field where it's going to be the worst. And there's some reasons to think that, but actually the fact that quality here is so observable, I would think also makes it harder. It's a disincentive for people to do this. And so people have asked us things like, why don't journals put floors on the quality that they'll accept? I think my hypothesis is that this again speaks to the importance of coordination. Projects have two dimensions in our model. There's how well they're executed and there's the importance. And, Cell and Nature and Science, they're very competitive with each other. So if Cell says we don't take anything below this quality threshold, Science is going to be like, great, we're going to grab all those super important projects that you won't publish now. That's a win for us. And so I think with a policy like that, you're going to need some sort of cross-journal collaboration to make something like that work. But that's speculative on my part.